It is my great pleasure to welcome Dr. Rizwana Islam to the podium to present their latest optimization of GALAN-S enzyme, both activity and stability. She is a drug discovery scientist with more than 12 years of experience developing treatments for patients with rare and difficult to treat diseases, including rare metabolic disorders, lysosomal storage disorders, and neurological conditions. She earned her PhD in biochemistry from Harvard University and has held research leadership roles at Shire, Takeda, and now at Crosswalk Therapeutics in Cambridge, Mass., where she serves as Senior Vice President and Head of Biology at Crosswalk. She is pioneering a platform that delivers medicines directly to hard-to-reach tissues. She's driven by a passion for turning innovative science into real therapies for patients with few or no treatment options. Please welcome Dr. Rizwana Islam.
Hi, everyone. Thank you for giving me the opportunity to speak to you today, and thank you to the MPS Society to allow us to share some data that we have generated for new next-generation enzyme replacement therapy for MPS IVA or MPS. Sorry, bear with me. I'm just trying to figure out the... Okay, all right. Let's start with a quick overview of the disease. So MPS IVA is actually caused by, it's a rare lysosomal storage disease. It's caused by mutations in the GALAN-S enzyme, the lack of which results in the accumulation of these long-chain sugar molecules called glucosaminoglycans, or GAGs. And specifically for MPS IVA, the kind of GAG that accumulates, as you heard yesterday as well, is keratin sulfate and chondroitin 6 sulfate, which are mostly made in the cartilage and in the bone. The prevalence of this disease is less than 1 in 200,000, and it affects multiple organs within the body. So as I mentioned, these GAGs are produced in the bone and cartilage, so these are tissues that are strongly affected, as well as the heart, the respiratory system, the eyes and ears, and so forth. While there is an enzyme replacement therapy that is approved and that does show some clinical benefit, there remains an unmet challenge for getting the enzyme delivered to the bone and cartilage. And let me walk you through the biology of the disease and what happens a little bit more in detail. So in a healthy cell, the GALAN-S enzyme is found in the lysosome. The lysosome is the recycling center of the cell. And there, the enzyme cleaves off this chemical tag called a sulfate from the GAGs, such that they can be broken down and cleared from the body. In the absence of the GALAN-S enzyme, where it's mutated and not functional, these GAGs tend to accumulate, resulting in malfunction of the cell and complications that we see in the different organs occur. All right, so quickly giving you an overview of what we are trying to solve and why we're trying to solve it. So this is a common issue with a lot of the enzyme replacement therapies. All of these enzyme replacement therapies rely on replacing the native enzyme, which are lysosomal enzymes. And they're not designed by nature to be effective drug-like molecules. What happens is that when you inject them into the body through the bloodstream, these enzymes are lysosomal enzymes, meaning they're the happiest, which means most active and most stable in the low pH environment of the lysosome. In the bloodstream, in the neutral pH environment, they actually fall apart quickly and lose activity. They also are taken up largely by the liver and do not really get delivered to the bone and the cartilage as well. And repeat injections are often associated with immune responses that can be triggered. So how we have tried to solve this problem is that we have actually designed these engineered fusion enzymes that have a targeting tag that allows them to reach the diseased tissues much better, and rather than going just to the liver. These enzymes are also engineered to be much more stable and last longer between doses in the tissues where they're needed. And we'll also engineer them for better safety such that there's a lower possibility of triggering an immune response. All right, so how we have done this is, as I said, we engineered the enzyme itself. So in this case, for the GalNS enzyme, we have engineered it to be more stable, which means that the molecule falls apart less quickly and stays and is able to process more GAGs for longer time. And it allows better clearance of the sugars that build up in the cells. And we've added this homing tag, which is a small single-domain antibody that targets the transferrin receptor. You've heard multiple other approaches earlier today about targeting the transferrin receptor to target the brain. This receptor is actually found throughout the body. It's involved in iron transport, and therefore all cells need iron to survive. So we've actually leveraged this natural mechanism to drive better uptake into cells that are normally not well addressed by the natural mechanisms that are in place. So when we put these together, they work in concert to make a better enzyme that reaches the tissues where they're needed and lasts longer and works better while they are in those tissues. All right, so we have actually spent quite a bit of time to think about this platform. We have a number of these, what we call binders, that target the transferrin receptor. We have designed them such that they reach all of these hard-to-access tissues much better than the conventional ERTs. We also made them species cross-reactive, which means that they actually bind to the transferrin receptor from mice, as well as non-human primates, and of course to humans, which means that the path to development is much quicker. We can use the disease models without having to rely on special models for testing our enzyme, and we can use a quicker path to development in that case. We have also carefully designed the receptor binding antibodies such that they're completely safe, meaning that they do not interfere with iron transport itself. And finally, our flavor of these binders are really small. They can be attached to any cargo. In this case, it's an enzyme, without hurting the properties of the cargo itself. So the enzyme properties such as activity, stability, secretion, expression, and so forth are completely retained even after adding that fusion tag. All right, so now sharing some data that we have generated. This is not for MPS IV. We look at this as a platform approach, so we've actually tested multiple lysosomal storage enzyme diseases with this platform. Here's data from another disease. So as I mentioned, the TFR receptor is actually expressed all throughout the body in all deep tissues. And as such, while it has been used to drive enzyme exposure in the brain, and we see that too, we are actually seeing 2 logs higher, better biodistribution in the brain with our fusion molecules. We also see much better expression in the kidney, the heart, and most importantly for MPS IV, the bone and cartilage as well. All right, so what we have done specifically for MPS IVA disease is we combined these approaches. So we used AI-guided tools as a starting point to generate variants that have favorable characteristics, make the enzyme more stable, more active, better expression, and have lower immunogenicity. And we added our fusion tag such that it's better delivered into the bone. And then we combined these and tested them in multiple in vitro systems that we set up. And all of this was actually funded through a grant we received earlier this year from the MPS Society that enabled this work. We were very excited by our initial data, so we actually went ahead and ran an additional in vivo study in mouse to look at biodistribution and confirm that our enzyme is getting to where it's needed. All right, so while we had a long history of working on multiple rare diseases, we had not really worked on MPS IVA before. So in the last 6 months since we received the grant, we actually had to set up our whole toolbox to enable the program. So we started with ways to actually make the protein. We developed cell lines that co-express this additional enzyme that GalNAs needs to retain full activity. It's called SUMF1, or formylglycine enzyme, that adds this modification to the enzyme to allow it to have full activity. We also made improvements to our protein production capabilities while we were doing all of this work. We developed a number of assays to look at how well the enzyme is performing. So we, in terms of catalyzing the GAGs or breaking them down, so we developed 2 different types of assays to measure the enzyme's characteristics. We also developed methods to analyze the stability of the enzyme as well. And I'll show you data that speaks to that. Next, we wanted to look at whether these enzymes are better taken up in cells. And we actually developed worked with 3 different types of cells. So first, we obtained multiple cell lines from Coriell, which are MPS IVA patient-derived cell lines, and screened them for enzyme activity to see which one actually had lower enzyme activity and accumulation of GAGs. We also developed a system of chondrocytes, and we're actually introducing GalNAs knockout mutations in them such that that can become a system for us to test our molecules in. And finally, we ran an in vivo study. So we developed very sensitive quantification assays that could measure our enzyme in different tissues. And in anticipation of future work, we have developed immunohistochemistry methods that can help visualize where the enzyme goes in tissues after. All right, and I briefly mentioned the chondrocyte knockout cells, and why we're making this is because these are the target cells. Chondrocytes are cells that reside in the cartilage and are essentially the building blocks there. And we currently lack a good disease model to show that our enzymes work well and they do what they're supposed to do. So we essentially took bone marrow-derived stem cells, and we differentiated them into chondrocytes, and we're actually knocking down GalNAs expression in these cells. And this is work in progress, so eventually we'll look at GAG accumulation in these cells, but currently we've already used these cells to show uptake of our molecules into the cell system. So we're essentially developing a disease-relevant in vitro cell-based model. All right, so now jumping into the work that we have done, I mentioned that we engineered, so I'll talk about the engineering first. We're breaking this down into 2 different pieces. The first is the enzyme engineering. As I mentioned, we used generative AI tool to come up with a list of the variants that could be favorable for improving the properties of the GalNAs enzyme. And we then manually curated this set of mutations. So we excluded any residues that are important for the dimer formation of GalNAs. And the GalNAs actually exists as dimer and has full functionality in its dimer form. We included all residues that are needed for glycosylation, and we excluded any changes that would affect its catalytic efficiency directly. We also screened out known pathogenic mutations, and of course, we excluded those from the list as well. And then we sort of, from a long list of mutations, actually hundreds of them, we selected as a first pass the top 20 of these, made them, of course, these need to be validated through biochemical assays. And then we looked at expression, activity, and stability, as well as uptake. And then we were able to identify multiple variants that had better properties. So in our next round, we combined some of these, and we will show you data pertaining to those. All right, and while we were doing the enzyme engineering, as I mentioned, we had a platform that allows better targeting of molecules to different tissues. So we have different binder sets, and we took a few of them, actually 4 of them, and selected the one that pairs best with the GALN-S enzyme by essentially the one that gave us the best uptake into cells. And then we combined these two approaches to come up with our lead molecule sequence. All right, so here's the first set of data, looking first at the round 1 engineering. So I said we made 20 variants. As you can see, 17 of them actually had better expression than the wild type. The best one actually had 10-fold better yields than the wild-type GALN-S enzyme itself, which is this molecule E17 that's highlighted in yellow in the graph that you see. Next, we took the ones that had good expression levels, and we put them through our activity assay to look at how the activity compares to the GALN-S enzyme. And we were very excited to see that most of the ones that had better yields actually also had better catalytic efficiency as well. So again, the variant that I pointed out earlier, E17, also had the best catalytic activity. So here's sort of like a summary of our findings. So the 4 variants basically jumped up to the top. This is plotted with activity against expression, and the ones in the yellow circle show you the variants that had a combination of both good expression and good activity. There were some variants which, while we saw better yields, actually had lower activity, so we excluded those and proceeded on to make different combinations of these 4 variants. And when we made the combination, this is R2 engineered GALN-S, or we call it R2E GALN-S. And compared to the wild type, you can see that this retains more than 10-fold better expression than the wild type protein. And again, this is just the beginning. This is all work done in the last 6 months. We can continue to engineer and improve this much farther. There's more. This can go more. All right, so now going from the engineering piece to the delivery piece, as I mentioned, these are binders that we had spent the last couple of years developing. We took 4 of them that were actually pre-validated through earlier experiments and have all the desired properties that we were looking for in the binders. We paired them with wild-type GALN-S in this case, and as you can see, variant 4 actually gave us the best uptake in MPS IVA patient-derived fibroblasts. So we proceeded on to making our next set of fusion molecules with this particular binder. All right, and now we basically are going to show you data with what happens with the combination. So this is an experiment in mouse endothelial cells. We use this as a system because we were going to eventually run our in vivo studies, in vivo biodistribution studies in mice. And this is a good example of what we would hope to see. So as you can see, the wild-type GALN-S enzyme is taken up into these endothelial cells. When you add the anti-TFR targeting to the wild-type enzyme, you see a six-fold boost in the amount of enzyme taken up. Now when you add on top the engineering to it, you see a further boost. And together, it results in synergy. So you actually end up seeing a 10-fold better uptake into these cells compared to the wild-type GALN-S enzyme itself. All right, and then I spoke a little bit about how we are developing a chondrocyte disease model. These do not have the knockout yet, that is coming, but these are still chondrocytes, which is the target cells that we want to target in the human body. And again, our pattern of better biodistribution or better uptake into these cells persist. So we see that the wild-type GALN-S is taken up in these chondrocytes, but as soon as we add the anti-TFR and the engineering, we see a much better boost, a 2-fold better uptake into these cells with our engineered fusion enzyme. All right, and then we heard a little bit about the mannose-6-phosphate receptor earlier on in the previous talks. That is the receptor that allows uptake of the native enzyme into different cells. That is also the way most of these enzymes are largely taken up into the liver, which happens to be the main depot many of these ERTs get into with little left to go into other organs. So we wanted to see the contribution of the mannose-6-phosphate receptor versus the TFR in terms of uptake into cells and what they contributed to. So as expected, when you have an excess M6P added to the system, this competes with uptake through the M6PR. And with the native enzyme, you see a 60% drop in uptake, which is as expected. With our modified enzyme, the enzyme is actually taken up at much higher rates than the native enzyme, 6-fold higher to be exact. And when we add excess M6P to it, the drop in uptake is very minimal. So this shows that most of the uptake is actually being driven by the TFR and not through the M6PR. Just to clarify, we did retain all of the mannose-6-phosphates on the protein as of now. And so this essentially has dual mechanisms. The uptake happens through both, but this data shows you that the primary mechanism of uptake is through the TFR. All right, now looking at some of the biophysical characteristics of the enzyme itself. So this is now the data that I'll show you with our chosen anti-TFR fused to our round 2 engineered GalNAs enzyme. And what we saw, which is reiterated as before, our fusion molecule has better baseline activity than the native protein. But what was actually very exciting for us to see is that it retains this at least 2-fold better activity in different pH environments. And why this is important is because the lysosome is at a lower pH, and this is where the enzymes would need to be active in the cells in the human body. So at pH 5.5 and even below at pH 4, which are the pH is found in the lysosomal environment, our fusion molecule retains much better activity than the native protein, but it also retains much better activity in the native pH environment, which is the pH of the blood. So you could expect this enzyme to retain its activity and stability in circulation as well as in the lysosomes. And then next we looked at how how we have improved the stability of these enzymes. So the first thing we did is look at the melting temperature and see if we are seeing any improvements in the enzyme properties. And essentially the melting temperature is the temperature at which the enzyme falls apart and is unfolded and no longer very functional. So the wild-type enzyme has a melting temperature of around 70, and even with our first rounds of engineering, we saw about a 4 degrees improvement in the melting temperature for this enzyme. And again, as you can see, this pattern of better enzyme activity is retained across multiple temperatures. So 37 degrees is the temperature of the human body. So we would want our enzyme to have and retain more activity at that temperature. And that is exactly what we saw. And we also pushed the temperature much higher, closer to the melting temperature of 50 degrees, again, to look at stability of our enzyme. And what we are very happy to see is that this enzyme retains better activity at that 50 degrees as well. All right, so we were very excited with all the promising in vitro data that we were seeing. So we wanted to test this and see how this performs in an animal model. So this is a study that we conducted in wild-type mouse. This is not a protein study. What we did here is inject DNA or plasmids that encode or the 2 different proteins, either the wild-type enzyme or the fusion enzyme into mice. And the reason we used plasmids instead of protein is because this gives us a quicker path and a less expensive way to evaluate these proteins. What happens is that with this injection of DNA, you see a brief burst of expression mostly from the liver, that doesn't last beyond 24 hours. And while most of the production is happening from the liver acts as a depot of production, making and secreting the protein into the bloodstream, and then the blood then takes the protein to all the other tissues, which then take up the protein into them. So it's a good way to mimic what we would hope to see with an enzyme replacement therapy. What we see here is both and the DNA is given at the exact same doses. And what we see here is the wild-type enzyme and our fusion enzyme are both expressed in the liver as expected. And our fusion enzyme is more stable, so we do see higher levels in the liver. But if you compare the levels of the two, there are some animals, there are two dots. I'll try to use my pointer. All right, so there are a couple of animals that you see, they have similar levels of expression in the liver, but now when you look at the cartilage and in the heart, there's actually significantly higher exposure into both the cartilage and the heart with the fusion enzyme, and essentially nothing in the cartilage and very minimal levels of exposure in the heart. So not only is the engineering for better stability important, it's also the transferrin receptor targeting that drives the enzyme into these tissues, because there are animals where the levels are similar, but this animal doesn't have any exposure in the cartilage or very minimally in the heart. So overall, you need both aspects, better targeting and a better enzyme, to give us what we're hoping for. And even just with this starting point, we're seeing more than 10-fold better exposure in the heart and more than 50-fold. This is actually a divide by zero problem. It's actually much higher levels of exposure in the cartilage as well. All right, so to summarize it all, there remains unmet need, especially with targeting the bone, the cartilage, the heart, and other tissues that are affected in the disease. We are designing our next generation ERT to address all of these unmet needs. Essentially, what this means is that we've engineered it for better exposure to the disease, the tissues that are affected in the disease. And we have also made the enzyme much more stable, much more active, and have much better yields. And they're more stable both in the lysosome, meaning much more stable in the tissues where they're needed, as well as more stable in circulation and even for storage and so forth. We look at this as a platform opportunity. This same principle could be applied to multiple other diseases as well. To sum it all up, I'd really like to thank the MPS Foundation for giving us the grant to enable this work and move it forward. And I'd like to thank the Crosswalk team who had worked on this and made this data package possible. Thank you so much.
Thank you very much, Rizwana. That was an amazing presentation in a very short period of time. Questions for the audience?
Yes, it's impressive to see some of the effects on the enzyme. I'm still a little lost, and maybe all these things are relevant. When you talk about it being a better enzyme, you know, is it has a higher Vmax? Is it binding substrates better? To me, you know, and then in terms of expression, is it better codon utilization? When I look at the data, it really just seems like it's more stable, and that may account for most of the things. But without getting into which amino acids were changed, does it make sense when you look at those amino acids, the effect that you're seeing on the enzyme?
No, absolutely. Good question. So it's not just codon usage that's optimized. It's actually, we have changed specific residues. I haven't actually gone into how we came up with a set of variants. So this was actually done, we have actually done protein engineering in the past, and the current AI tools that we're using, we have actually validated the tools using feeding it into the data that we have generated from our other protein engineering platforms and making sure that the predictions that it makes are actually accurate. So accuracy is above 50%. Of course, it's not higher than that. And the next step lies in actually testing them. And the way it comes up with this list is looking at related enzyme across multiple different species, as well as multiple different closely related enzymes, and coming up with changes or variations that could be favorable instead of deleterious. So the point mutations that you're seeing, these are not just codon optimization. This is actually changes in the amino acid sequence.
That makes sense. And we were just saying, I mean, we think, nature made these enzymes perfect, but maybe nature, Dr. Munzer and I were saying, maybe nature didn't do as good a job as we thought,
Other questions from the audience?
Very, very exciting. The current standard of care for MPS 1, 2, 4, 6, and 7 does not reach the bone. You mentioned at the end of your talk that this possibly could transfer to other MPSs. And Dr. Steed's point, one question I had was, is it possible that the enzyme is not actually better, but the uptake is so much more immense because it's not just mannose-6-phosphate, but TFR? So both of those questions with regard to uptake and also how long, there's a line to get to the clinic. So what is the route so families will understand great science before it actually gets into patient?
Absolutely. So like I said, we have just started. So the next steps would be to validating this enzyme in a disease model. And then the next step would be to run a safety study in a relevant species and essentially prepare a data package that would be IND enabling. And then we can plan for a phase 1 proof of concept study in the clinic. So like I said, it's a 6-month-old program, but there is further validation. We have used the same platform in our previous lives for Hunter. That was actually done in a gene therapy context. And it's sort of like a little bit of a trajectory, but as you can imagine, the cargo that we're making can also serve as a cargo for delivery through a gene therapy as well. So we have actually published a lot of data around this platform as a concept from our previous lives and tested this out. And as you mentioned, there is better uptake of the enzyme into different tissues. It's not just one tissue. The TFR is everywhere. So you get better biodistribution in the brain, you get better biodistribution in the bone, the cartilage, the heart, and so forth.
Do we have other questions from the audience?
So I know it's very preliminary, but if this was to be available to patients, how would this figure into our current enzyme replacement therapy that we're currently receiving? Would this be an addition or would this be a replacement? How does that work?
So we envision this as a better treatment for MPS IVA disease, where it can address some of the concerns that are not currently addressed by the current ERTs that are available. So we would expect better outcomes for the bone. We would expect better outcomes for the heart and so forth. Oh, so we are primarily exploring IV at this point. We have tested subcutaneous delivery for another disease, but again, we haven't done that yet at all for this indication. So we would see how well this protein and this fusion, is this amenable to sort of concentrating it into a small volume for subcutaneous dosing and so forth. So research needs to be done, but that's one possibility, where it could be less frequent IV dosing, or it could be subcutaneous dosing to be explored. To be seen. It's a similar mechanism as vimism with add-on properties that allows it for better delivery to other tissues.
And how would you foresee your next steps after you get an optimized enzyme that works really well in the in vivo models? are you planning to do just a protein delivery?
Yes.
Or, oh, okay.
That is essentially the next step. So we want to make sure that and we have, again, used this approach for multiple other programs. What we see is that this is sort of like a quick and dirty experiment, for lack of better words, to mimic what would happen in an ERT. The reason we did an ERT right away is because it takes highly pure protein, much higher volumes of protein that we would have to make, better quality, so it's more expensive and takes longer to do that. That's our next step. Confirm that we see the exact same biodistribution pattern in animals with the protein.
Would you also envision delivering as a gene therapy vector?
We currently don't have the means to do that, but that absolutely. So this could be a viral or a non-viral cargo for a gene therapy delivery platform. Yeah.
Any other questions for the audience? Getting my steps in.
Running. Rizwana, so why did you go first with 4A and with the mouse model? Is that where you're going to go next with that? Because I think Dr. Munzer mentioned yesterday there's not as much of the bone disease in the 4A mouse model.
We actually focus on where there is unmet need. So there's a lot of programs that are being developed for Hunter, for Hurler, for many of the other MPS diseases. So we've looked at where there isn't a lot of research on right now, and then that's why we focus on MPS 4A. But you're right, this method could be applicable for many of the other indications too.
I wanted to let you know that the National MPS Society has funded the creation of an MPS 4A pig that is available, and if you didn't know about that, that might be a useful model resource to evaluate this novel therapeutic.
We would love to. Thank you for that.
Find me after the talk.
We would need to test the cross-reactivity of our binder to pig TFR, but if it is, then absolutely.
Excellent. Well, if there's no further questions, I want to thank Dr. Rizwana Islam again.